Hello students, Assalamu alaikum. Hope you all will be fine. Let's start today's lesson that is about cell cycle. According to the cell theory, new cells originate by division in the pre-existing cells. The cells which divides is called the parent cell and the new cell formed as a result of division are called daughter cell. The process of cell division is needed of development, growth, healing and for sexual and asexual reproduction. Cells not only increase in number but also manage to transfer genetic characteristics to the next generations. During cell cycle, cell grows in size, form new molecules and organelles, replicate their chromosome, and divide by equally distributing genetic material in the daughter cell. The cell cycle is a sequence of events which involves growth of newly formed cell, replicates its genome, and divides into two daughter cells ultimately. It consists of two main phases, interphase and mitotic phase. Interphase is the period in cell cycle between two consecutive divisions. It is divided into G1, S, and G2 phase. Interphase is a period in cell cycle between two consecutive divisions. Interphase is the longest phase of the cell cycle, during which the cell grows, carries out its normal functions, and prepares for division. Interphase is essential for cell growth and genetic material duplication before mitosis or meiosis. It consists of three stages: G1 phase, S phase, and G2 phase. Interphase starts with the production of new cell. Cell grows in size, increase the number of its organelles, forms needed proteins and other substances. During this phase, cell prepares itself for the next phase of cell cycle. It forms enzymes and nucleotides of DNA required for replication process. S phase is the synthesis phase of DNA. The cell replicates its entire genetic material to form its two copies. That is why each chromosome has two chromatids during cell division, one for each of the daughter cell. These chromatids are attached with one centromere and are called sister chromatids. During G2 phase, cell prepares for mitotic phase. Cell forms proteins, especially those required for the formation of spindle fibers. Also, cell accumulates energy to complete the division process. More mitochondria are formed in this phase. During G1, cell may exit the cell cycle and enter in G0 phase. In G0, cell stops to divide. Some cells, for example neurons, once mature, never divide again. They remain is in G0 forever. Cell of liver, kidney, etc. enter in G0 temporarily. When needed, they re-enter the cell cycle and start to divide again. Many epithelial cells divide continuously. They never enter in G0 phase. Next phase is mitotic phase, in which one parent cell is divided into two daughter cells. The cell division in which parent cell produces two daughter cells with the same number of chromosomes as in the parent cell. Mitosis is a continuous and very fast process which takes less than an hour. But to study easily, it is divided into karyokinesis and cytokinesis. Karyokinesis is the division of nucleus which form two nuclei in a dividing cell. It is further divided into prophase, metaphase, anaphase, and telophase. During interphase, hereditary material is formed in the form of very thin threads called chromatin. If a cell is going to divide, all of its chromatin fibers duplicate during S phase of interphase. Prophase is the longest phase of cell division. At the start of prophase, chromatin fibers coil up and condense into chromosomes. Due to duplication, all chromosomes consist of two chromatids. The chromatids of each chromosomes are attached to each other at centromere. Nucleus disappears as its DNA is packed into chromosomes. At the end of the prophase, nuclear membrane splits into vesicles which disperse in the cytoplasm. Spindle apparatus is formed. In metaphase, chromosomes are aligned at the equator of spindles. Two spindle fibers, one from each pole, are attached at the centromere of chromosomes. In anaphase, spindle fibers pull the centromeres, which split and chromatids are separated from each other. Individual chromatids are pulled until they reach their respective poles. In telophase, chromatids reach at their poles. They uncoil and lengthen to form chromatin fibers again. The spindle apparatus disintegrates. Nuclear membrane is formed around chromatin at each pole. Nucleoli reappear in both nuclei. Cytokinesis is the division of cytoplasm which results in the formation of two daughter cells. It starts while telophase is in progress. The pattern of cytokinesis is different in plants and animal cells. Cell membrane in animal cell begins to invaginate in the equator region. As a result, a cleavage furrow is formed which continues to grow inward. Cell membrane in the furrow finally join up and separate the two daughter cells. In plant cells, spindle fibers in the equator region form a structure called phragmoplast. Golgi operators form vesicles which appear in the center of the phragmoplast initially and then grow at equatorial plane. The content in these vesicles form middle lamella and primary walls of the daughter cells. Later on, some cells form secondary walls. The main function of mitosis is to maintain the number of chromosomes and genetic material in all cells of an organism. Following is the significance of the mitosis, development and growth. Life of sexually reproducing multicellular organisms starts from a single cell, the zygote. The continuous cell division process leads to the development of adult form. The organisms also grow in size by increasing cell number. Thus, the development and growth of multicellular organisms depends on mitosis. Cell replacement and wound healing. The replacement of worn out cells involve mitosis. The epithelial cells of skin, digestive tract, and respiratory tract die off regularly. They are replaced by identical cells produced by mitosis. Damage repair and wound healing depends on the process of mitosis. Mitosis is the basis of asexual reproduction. This reproduction involves only one parent. All asexually produced offsprings are genetically identical to their parent organism. The vegetative propagation is very common in plants. It involves new generations from root, stem, or leaf of parent plant. Potato, onion, garlic, ginger, grasses, etc. reproduce asexually. Many animal species like sponges, planaria, and hydra undergoes asexual reproduction. Genetic stability. Mitosis produces two daughter cells with the same number of chromosomes as in parent cell. Before the start of cell division, DNA of the parental chromosome replicates to form two exact copies. Daughter cells thus will have same genetic information. That's all for today. Watch the next video to learn about the meiosis. Like the video and subscribe the channel for more videos.